Good evening. Okay, so in our last class, we have almost completed everything that we had to discuss regarding the B cell development, maturation, activation, all the processes by which the B cell receptors are rearranged, how the diversity component is attained, and all the relevant notes to your B cell development and everything has already been shared with all of you now. So for today's class, we will just complete one last topic that we could not manage to finish the last time, which is the process of selection that the B cells undergo. Because once a cell is getting rearranged, going through the process of rearrangement of its receptor, we have briefly discussed this previously that it might be there might be a possibility where they develop certain antigen binding sites where the receptors are actually recognizing your own body or self antigen. So we'll see how the elimination of those cells happen, and then we will move to the discussion on T cells. So just 5 minutes more and we can get into our class for today. If there's anything, Shamika, from the previous class, then we can take that up in the meantime. Okay, so we discussed that we stopped our discussion at the point of somatic hypermutation, and this one graph here. So this one's discusses the fact that so this is the region of VDJ, and here are is the constant region, and there's a stretch of nucleotides which is in between these two genes, which will eventually get rearranged to bring about the entire functional heavy chain of your antibody molecule. So this over here is from the somatic hypermutation that we have been discussing, and it says that the frequency of mutations increases in the domain of this VDJ. And as the distance from this VDJ region increases, the frequency of mutation also goes down. So, as a consequence of all the rearrangement that takes place and then presence of antigen, this somatic hypermutation that is undergoing, all of this is going to generate a lot of different possibilities for the antibody molecule to recognize different antigens. But to prevent for any self-reactivity of the B cells against self antigens, which can be very dangerous for your own system, we have the role of selection that comes into play. Now, where is this hypermutation taking place? It was taking place in the dark zone of germinal center. And if you remember, the centroblast uh, ch- divides into, differentiates into centrocytes, which then migrate into the light zone of the germinal center, which is more towards the outer cortex of cortical region of your developing site, which is your secondary lymphoid organs. Now, in the light zone, which we have the somatic hypermutations, accumulated mutations, and all those cells coming in. And there is two possible ways. One, that we select for the combination that is generated of the for the antibody molecule, which means that whatever is the reactivity, whatever is the antigen specificity of the receptors on it, we can have the cell selected for, or the contrary possibilities that we signal that the cell is self-reactive, it can be dangerous to the body, and such cells are taken for negative selection, right? Now, in there must be all of these three signals that you see here, these all needs to be ticked off for a particular cell to undergo positive selection or a favorable selection. So what is it? We should have that whatever is the recombination of heavy and light chain, the VDJ recombination that happens, the combinatorial arrangement of the heavy and light chain amongst itself, whatever is the ultimate consequence of it, if I have a expressed antibody molecule that can bind the antigen which is displayed on the follicular dendritic cells. So I'll bring you here where in the light zone we have dendritic cells, the follicular dendritic cells, which on its surface, these small red dots that you see, these are all antigen coated surface of the follicular dendritic cells. We have discussed this in our previous lectures, the very initial lectures of B cell development. So the first selection signal comes from that the ultimately expressed antibody molecule is able to bind the antigen which is being displayed on the follicular dendritic cells. Next, so also what happens is it again involves the next signal again involves the follicular dendritic cells, and what happens is that since we have a limited amount. So here you might feel like there's a lot of antigens which is coated on the surface of your follicular dendritic cell. But it is also important to recall the fact that not all of these antigens, which is exactly being presented by this follicular dendritic cell, is the same type. We have this follicular dendritic cells which might be presenting different antigens. So maybe that this B cell that is recognizing these two antigens is of a different specificity because these antigens is of a different make, whereas a B cell here is recognizing some other antigen. So it is important to understand the fact that this follicular dendritic cell is not presenting one antigen at a time, right? It is presenting every antigen that it can possibly present on its surfaces. So you would see that the entire surface is coated with antigen, but every antigen is not the same. Now, okay, so let's just say that while all the antigens are red, but right now what we are interested in is this blue color antigen. So the frequency of this blue antigen being presented on the follicular dendritic cells is very less, that we have this one particular antigen against which I need the centrocytes. So now all the centrocytes that are interacting with the follicular dendritic cell in the germinal zone, they will actually fight or compete amongst each other to get a chance to bind to this blue antigen. Now, so that is your second signal, that whichever centrocyte succeeds this competition of binding to the limited supply of antigen being presented by the follicular dendritic cells. How is it succeeding this competition? Is by having the highest affinity. So if you see here, we already had somatic hypermutation going on inside the dark zone. Because of somatic hypermutation, there is a process called affinity maturation, which the centrocytes are undergoing. Now, because of affinity maturation, the highest affinity centrocyte needs to get selected. This selection is being possible because there is a competition amongst the centrocytes to bind to the limited supply of antigen being displayed on the surface of follicular dendritic cell. The possibility is that this centrocyte here is also in close vicinity of a blue antigen that is of interest to us right now. But because the affinity of binding of this interaction might not be so strong, so we will eventually have this particular centrocyte falling off this interaction, whereas this centrocyte continues to have its chance of interacting because the affinity of this binding is very high, right? So that is the second selection process where highest affinity centrocyte is getting selected. And how is it becoming a possibility is because of this limited supply of antigen. The third signal for positive selection is the interaction of CD40 and CD40L molecule. So if you would remember again from our previous classes, that CD40 is a molecule which is present on the B cells and CD40L is present on the T helper cell. Now for T helper cells activation, the interaction of CD40 and CD40L is it's a signal for the activation of T helper cell. But it's not just this T helper cell that requires the interaction of CD40 and CD40L. It is also the requirement of B cells, which is expressing CD40, to interact with the CD40L molecule present on the T helper cell. Why so? Because this interaction of CD40 and CD40L is going to give a stimulatory signal for T helper cell activation, which in turn is going to release certain cytokines. The cytokines will further select for the B cells that is going to take care of the antigen, right? So CD40 and CD40L of the T helper cell interaction is required in addition to the processed antigen MHC complex interaction that is required by the TCR, but it is also required by the BCR for its selection through the process of positive selection. Now, if either of these criteria is not being ticked off by the centrocyte that has formed after all the recombination and somatic hypermutations, such cells is going to undergo apoptosis, right? So, which all cell gets into apoptosis? One, if it can bind to a self antigen. Second, if as a consequence of all the hypermutation that it accumulates, the binding affinity is either of the same strength or if the strength is reduced. So in either of these cases, we will have the cells getting triggered for undergoing apoptosis within these germinal centers itself. Because if you see, everything is actually happening inside the germinal center, either in the dark zone or in the light zone. Now, so we say that they are getting selected for apoptosis, but but before the cell actually is signaled for apoptosis, it is actually getting a chance to undergo the process of receptor editing, or you can say that before the cell is signaled for death, there is there are other processes that happen in between, and it's not really like one after the other. It is not very clear that which process precedes the other one and which one is a consequence of the other one. That part is not very clear to the scientific community yet. But the processes that mediate the self tolerance, or the processes, so again, self tolerance is the concept where the self antigens are not reacted upon by the B cells. Now, if we do have a cell that is non self tolerant, which is having receptors that can bind self antigens, such cells can undergo either of the three processes before the ultimate fate of it getting into apoptosis. The first one being clonal deletion. So it's like, so these processes are basically a marking before a cell is taken into apoptosis. So you can have a particular B cell to be marked for clonal deletion, wherein you have the self-reactive B cells which have to be deleted, right? Deleted मतलब इसका और कुछ नहीं हो सकता. It is a very strong affinity to self antigen, and this cell has to undergo death. But prior to the death consequence, the cells might, the ones that are binding to self antigen with some low affinity, it can be, those cells are given a chance to undergo the process of receptor editing, wherein you replace the self-reactive receptors. So here you have the entire self-reactive B cells which are taken for death. However, in receptor editing, we have the self-reactive receptors which can get replaced with new receptors. And how will that be possible? Again, from our previous classes, we know that we have two copies of our so two alleles for your all the heavy and light chain genes, and only one of them first enters the process of recombination, right? Now, if we have one of the recombination that happened with one of the alleles resulting into a self-reactive BCR, we can have that so it's the cytokines in the body. This signal for these cells to again re-express the RAG proteins and enter the second round of BDJ recombination. So here the receptors get another chance to form altogether, wherein a new antigenic specificity will arise as a matter of chance, right? So after the recombination, second round of recombination, if now we have a productive recombination, or we have the antigenic specificity which is of a higher affinity to the antigen in question, those cells can get selected for. But if again, despite of receptor editing, we again have a self-reactive or a lesser affinity PCR, those cells can again be coming into the situation where they have to undergo clonal deletion. The other possibility, the mechanisms of which is not very clearly understood, is the process of clonal energy. So energy is that here the cells are completely deleted off, but in energy we have functional inactivation and then subsequent cell death. So subsequent cell death is happening in both these consequences, right? So the first chance, the only place where we give a chance for second round of rearrangement is the process of receptor editing. And that is also not a case that where the cells get the chance of getting into second round of recombination all the time. That also again has selection criteria, and depending upon the cytokine milieu in the environment, the cells can get into receptor editing, right? So we either have cell death. If everything fails, we have cell death. Or if the cell continues to survive, we have clonal energy. In clonal energy, whatever is the product of this self-reactive B cell, so the antibodies that it produce, they can be functionally inactivated, right? So just as say that if this is a self-reactive antibody, we can have maybe some other antibody taking care of this particular antibody and resulting into functional inactivation of your products from your self-reactive B cells, right? And eventually, when these cells are not, because eventually every cell has a shelf life, so eventually those cells are anyway going to be cleared off, right? Because once we have a B cell which is self-reactive, before it can be taken to that, the products from it are anyway undergoing functional inactivation. So these three are the mechanisms of negative selection in a way, and it is for all the B cells that can be self-reactive. Now, where is this education of self and non-self antigen happening? Again, it is happening during the process of maturation, where we have the follicular dendritic cells purposefully expressing or presenting the self antigens to the B cells, and any B cell that reacts to these self antigens are undergoing these processes. Only the selected population of B cells are allowed to come out and reach into the peripheral circulation. Okay, so the diagram here denotes the same concept that we have just discussed, wherein we can. So in central tolerance that you see, this is so these processes of receptor editing or clonal deletion is happening in the central lymphoid organs or your primary lymphoid organs. But if we still have some cells escaping this process and reaching the peripheral circulation, which is your peripheral lymphoid organ, we can again have certain process of selections to eliminate the self-reactive B cells in your peripheral lymphoid organ. So the process of selection in your peripheral lymphoid organ is primarily by the process of clonal energy, whereas the receptor editing and clonal deletion is the primary process inside of a central lymphoid organ. So with this, we complete our discussion on B cell. Everything about it has been discussed, and I'll just take a minute or two to read out the entire summary from the book, right? This will help you to also recall every concept that we have discussed so far. And if anything is not clear or you're not able to comprehend the entire picture in any possible way, you can we can just have a quick discussion as I'm reading out the summary for you all, right? So we saw that the B cells are developing in the bone marrow and they undergo antigen induced activation and differentiation in the peripheral organ. Now we can have this. What do the activated B cells do? They give rise to antibody secreting plasma cells, or they can differentiate into memory B cells, right? Now, during the process of, so this is what the B cells do. Now we are coming to the in detail process. What all are the things that is happening during these processes, right? So during B cell development, we can have a sequential IgG gene rearrangement. What does the word sequential mean? The word sequential means that one by one, the heavy chain genes and then your light chain genes are getting rearranged. This rearrangement is actually transforming your pro B cell eventually into an immature B cells, which only express membrane-bound IgM molecule with a single antigen specificity. So as a consequence of all this gene rearrangement, we only have B cells which can have a single antigen specificity. It's never like we have multi-specific or poly-specific B cell. All the B cell receptors are monospecific, right? So every cell can only recognize one particular type of antigen. Further, we have from the immature B cells, which are antigenically committed, they get into further process of differentiation, and we have mature but naive B cells, which are expressing both membrane-bound IgM and membrane-bound IgD. This self-reactive, if a self-reactive BCR is expressed in the bone marrow, there is a process of negative selection for the self-reactive immature B cell. The selected cells are deleted by apoptosis or undergo receptor editing to produce non-self-reactive membrane-bound IgM molecules. Now, B cells that are reactive to self-antigen in the periphery are taken care of by the process of clonal energy. We just discussed this. In the periphery, the antigen induced activation and differentiation of mature B cells. This is going to eventually generate antibody response, where most of this antibody response is requiring your T helper cells. So there are so because of the involvement of T helper cells, we also discussed very beginning of our discussion with B cell that the antigens that are stimulating B cells can also be of two type. It can be a thymus dependent response, or it can be a thymus independent response, right? So there are two kinds of antigen present in the environment. So in thymus dependent responses, we usually have the proteinaceous antigen that takes up a role. However, there can be some poly uh, polymeric peptides, right? So multiple repeats of the same protein that is joined together into a polypeptide, such sort of structural things, and the like the lipopolysaccharides and polymeric molecules, which have these repeated epitopes, they do not require the T helper cell presentation, and are therefore known as thymus independent antigens, right? But mostly the antigens that we encounter are thymus dependent. The next is that this B cell activation is ultimately a consequence of signal transduction, that does not only involve the B cell receptor or the antibody molecule. So B cell receptor and antibody molecule is the same thing. It's just that antibodies can also get secreted, but the bound form of antibody can also be referred to as the B cell receptor. So B cell activation is a consequence of signal transduction, and it does not only involve this B cell receptor, it also involves a lot of co-receptor molecules, right? We saw there is some alpha beta molecules, which were the signal transduction molecules. And why so? Because the C-terminal domain, which is inside the cytoplasm of an antibody molecule, is very small. So this doesn't have functional motifs, which can allow certain signaling molecules to interact with it. So whatever is the antigen binding to the B cell receptor, we have associated signal transduction molecule that pass on this information to the inside of the cell, where the different transcription factors can get expressed and bringing about a change in the expression of proteins and everything. So those are the functional responses, right? So B cell activation is a consequence of signal transduction process triggered by engagement of B cell receptor. So while we are saying B cell receptor, we are also referring to all the co-receptor molecules that are decorated along with the B cell receptors, right? Which ultimately lead to the many changes in the cells, including change in the expression of specific genes. Okay, so B and T cell activation, they also share there are many parallels, and we will discuss all these parallels in today's and the next class. I'm not sure why I am not able to. You all can hear me properly, right? There's no network issues from my end. Okay, Rajkumar is finally admitted. Okay, so there are many parallels of B and T cell activation, which includes that there is something called as compartmentalization of function within receptor units, that we have activation by membrane associated protein tyrosine kinases or PTKs. We will discuss the role of PTKs in today's class as well. And there is recruitment of several signal transduction pathways, right? Again, B cell co-receptors can intensify the signals resulting from cross-linking coming about by binding of the antigen to the IgM molecules. Right. So activation induced by TD antigens require a contact of CD40 on B and CD40L on T helper cells. Okay. So again, from thymus dependent and independent antigens, the thymus dependent antigens require interaction of CD40 of B cell and CD40L on the T helper cell. This is also an essential signal for survival of B cells. We just discussed this right now. And their formation of germinal center, the generation of memory cell population, and somatic hypermutation. So all these phenomena, so formation of germinal center, survival of B cell, generation of memory cell, and somatic hypermutation. So all the important processes that are actually helping to bring about a sufficient immune response of the B cells, they are all dependent on the interaction of CD40 and CD40L. Now, we also discuss something called as a primary and a secondary antibody response, and we understood that they are different from each other. So, primary response has a long lag period, whereas uh, there's a long lag period, then there is antibody formation, a small plateau, and ultimately the peak decreases. IgM is the main antibody that is formed in your primary response, right? But following to this, there can be a switch to other classes such as IgG. In secondary response, the lag period is very short and there is a more rapid log phase. So its lag is very short and the height of response that you see is also very sharp, right? It's pretty heightened and it happens quite fast. And then there is a longer plateau phase. So in your plateau phase essentially denotes that how long your antibody remains in the circulation before their numbers eventually decrease. So there's a longer plateau phase and a slower decline than what we see in the primary response. And IgG is the main isotype. Along with the other isotypes, they can be produced in your secondary response rather than IgM. And the affinity of antibody produced in the secondary response is also higher, right? And so the germinal centers that form, which are the sites for somatic hypermutation and rearrangement of immunoglobulin genes or affinity maturation, all of these things are getting formed in response to your TD antigens, and eventually you will have memory B cells, class switching, plasma cell formation. So this is exactly, it's a very nice summary for what we have been discussing for the last 3, 4 classes. Take a minute to understand if there is anything that is not clear to you. Today's class is the following few pages of this is going to be comparatively easier for us to understand. It will also not be taking us a lot of time to go through it because we have already discussed the very similar mechanism that is happening in the B cell. So in T cell, you will always find a comparison between what you have already been studying about since the last 3, 4 classes. So we can go slow. We can try and while we are trying to draw parallel of the T cell with the B cell, it is also your chance to recall everything that we have discussed in B cells because eventually the two things will only come together. And since we have discussed in great details about B cells, today's class is going to be simpler for you if you all have followed me through these days. Just go through these lines once again. And if there is anything that you're not able to recall and you would like me to quickly go over it again, please stop me right now. In the next 2, 3 minutes, we'll move our discussion to T cells. So those of you, the new names that I do see in today's class who were not there in last class, I hope you all have followed through the recorded lectures or the live sessions that are being uploaded to YouTube for reference, wherein we have discussed some of the concepts, the names of which are are going to keep coming upon from the classes henceforth. So I hope you all are able to follow these terminologies and are not lost in the details. Can you please make a diagram which we have studied? We we did discuss everything in diagram, and I also said that once we are done with today's class, uh, primarily we are done discussing T cells. We will compile everything. I'll try to take that much time and compile all the information into one diagrams. But for now, if you need one reference diagram, I'll ask you to follow this diagram, because over here we have almost introduced every term that we have. So the following classes from here have only discussed about all that, how all these particular terms are coming into picture, right? But if you look in this carefully, you will encounter every process that we have discussed so far onto this backbone diagram, right? But hand-drawn flowchart or something like that, I will try my best to provide it to you. But one that I can only do once we have completed our discussion with T cells, because I'll try to bring the two cells also together. Okay, should we go ahead with this?
Excuse me, ma'am.
Yes.
Ma'am, I'm having some few
Yeah,
doubts.
yeah,
Can you
yeah.
ask
Please
me?
tell me.
Ma'am, in today's discussion, you have told that one B cell receptor encounter many antigens. So, in that case, how it is going to be specific for one antigen?
See, it's not like, so the follicular dendritic cells are presenting multiple antigens. The chance of different B cells interacting to this different antigens on the follicular dendritic cells is equal, right? So we have, um, say we have B cells which can be named from A to Z. We can have the B cell with name A recognizing antigen small a. We can also have another cell, B, B tagged B cell, which can also recognize this small a antigen, but the binding affinities are definitely going to be different. Whichever of these two cells have a higher binding affinity is going to get selected in the process of selection. But it will never be like we have a capital A B cell which was having a specificity for small a antigen to now undergo such rigorous mutation that the antigenic specificity has only changed. So this capital A B cell is committed to recognizing small a antigen. It will never go and recognize small b, c, or d antigen. But we can have a capital D antigen, which is specific for the small d antigens, which is also being presented by the follicular dendritic cells. So your follicular dendritic cells is presenting every antigen that is there that has to be taken care of, right? So it can present all A to Z antigens. You will only have B cells which can recognize either of these antigens. But there can be multiple A cells, right? It's not like we only have one A named B cell. Because of proliferation, we can also have two, three, 10 such sister cells which are having the exact same specificity for small a antigen. So the competition is also amongst all these A cells, and it is also the A cells competition with the B cells, which can recognize the small a antigen but with a lower affinity. Is this clear?
Ma'am, I have one more doubt with the example
Mm-hmm.
that you have taken. Like, for example, if a B cell is you have tagged as A is for small a antigen. Now, if that same B cell is expressed to the another, let's say small b antigen, then in that case, that B cell will be going to get antigenic specificity for that
No,
small b antigen, or that will be only for small a only.
it will only be for small a. That is antigenic commitment. So B cells are antigenically committed during their process of development. It will not be able to recognize a small b antigen because it is committed to recognizing small a antigens only.
Okay, ma'am. And ma'am, in the diagram that you have shown, in that the B cell receptor, there are multiple receptors that are present on the B cell. So that one B cell receptor is bounding to the one antigen, and the another is to the another antigen.
No,
So in
no, no.
that case, also the antigenic specificity is only
Yes,
there.
yes, yes, yes. So it is. So it's not like if I have said, let's just focus here. If I say that this arm of this particular B cell receptor is recognizing the blue antigen, this particular arm is also only recognizing a blue antigen if it is present. Otherwise, it is not going to interact with it. Similarly, the other B cell receptor, which is also present on the same cell, will only recognize it if it or bind to the antigen only if it's a blue antigen. If instead of it, it was a red, green, or any other antigen, because all the receptors on this one B cell is of the same specificity. That is why every B cell is monospecific. It is only going to recognize one particular type of antigen, which might be presented on the follicular dendritic cells multiple times.
Okay, ma'am. And ma'am, while saying or explaining about this, you have told that the antigen which is expressed in less amount. So what about the one antigen that is expressed in more quantity on the follicular dendritic cell? The same
Then
concept
we
will
will
be applied.
Yes, the concept remains the same. If I have more expression for a particular dendrite or for a particular antigen on a dendritic cell, then we will have more number of centrocytes which are going to get selected for. But which centrocyte? It's not like A, B, C, D random centrocytes are going to recognize those antigens. The chances of that is very less. It is most likely that the daughter cells of one particular centrocyte which got activated because of this one primary interaction can now come and recognize that same antigen presented on the follicular dendritic cells. Now, if we have increased number of this antigen being presented, we will have more number of centrocytes coming into picture so that the antigen can be taken cleared of.
Okay, ma'am. And ma'am, what is the difference between the role of maturation and the selection?
So in the process of maturation, we have the B cell receptors that is undergoing rearrangement and ultimately assembly. So we have initially have this Ig instead of Ig alpha beta co-receptors, we have some precursors to these co-receptors and all the assembly. So initially we have IgM only, but in a mature B cell, in all these processes of your expression of IgM, IgD, alpha beta co-receptor molecule, all of these things have already occurred. So such cell is known as a mature B cell. But you asked me what is the difference between a mature B cell and
We say that the maturation of B cell is taking place in the bone marrow, and
Mhm.
the selection word we have also come across. So, what is the basic difference between the maturation and the selection process?
Selection is happening inside your germinal centers, right? So the maturation process now, this is a good diagram to understand your question, where we have the pre B cell and the immature B cell, all of the pre B cell and immature B cell. And if you look here, we have all of this undergoing in the bone marrow, which is an antigen independent phase, right? But inside the bone marrow also, we have stromal cells, of which your dendritic cells and certain other antigen presenting cells is a part of that stromal network. So, because of self antigens being presented to these developing B cells inside of your bone marrow, we have a selection for self-reactive and B cells inside the bone marrow network as well. But the point, we have these mature B cells. So these are mature but naive because they have not encountered an antigen. So when these immature naive B cells reach the peripheral lymphoid organ, where they encounter this antigen. But now you have to recall that in presence of antigen, there is clonal proliferation undergoing, and whenever there is clonal proliferation, entire of this process is repeated, because in clonal proliferation also, this entire whatever is the BDJ combination, the daughter cells also has every possible other gene. So which genes ultimately get rearranged in this is going to be the same, because it is a clonal proliferation and BDJ combination remains the same. But in presence of antigen, hypermutations can take place. Because of such hypermutation, we can have a higher affinity, lower affinity, or maybe the chances of it being very less, but maybe a self-reactivity coming into picture. There, we mostly have clonal energy where the self-reactive products are eliminated, or if the affinity of binding to antigen is lesser now, the cell can get into apoptosis. So that selection is happening in periphery, because somatic hypermutation is a phenomena that is occurring in presence of antigen, and this encounter of antigen is only happening in the peripheral lymphoid organs. While in the bone marrow, in the antigen independent phase, we have the development where VDJ combination is getting fixated, because of which antigen specificity is fixated. So the selection for self-reactive cells, or the process of receptor editing and all those things, they are happening until the time the naive B cells leave to reach the peripheral lymphoid organs. In the peripheral lymphoid organs, because of somatic hypermutation, a process of selection is again required. So there we mostly have clonal energy or clonal deletion that takes care of that process.
Okay, ma'am. And ma'am, in the clonal energy, you have told that products are taken. So if they are taken and undergoing recombination, so a recombination or any use, so without cell, how it is possible?
Products are, so we have this secreted product coming out already. Recombination and everything is something that is happening inside the cell nucleus. So recombination happening inside of a product is impossible, because जो भी बनाया सेल ने उसको बाहर सर्कुलेशन में डाल दिया. अब उस प्रोडक्ट में कुछ चेंज नहीं हो सकता. But the binding of this product to the effector sites, maybe a antibody has been released by a self-reactive B cell, which can bind to a self-antigen, right? Self-reactive B cell is triggered for cell death. But जो उसने product बना दिया, उसका क्या करे? तो body tries to neutralize it or functionally inactivate such products by certain other mechanisms that take care of it. So maybe antibodies are protein molecule. Maybe we can have some protein protein esterase or some disulfide breaking enzymes that can cleave the disulfide in this self-reactive antibody molecule, and eventually the functional product is inactivated.
Okay, ma'am. And ma'am, one last point that you have
Mm-hmm.
drawn the tolerance section. If foreign is equals to acquired tolerance, so what does it mean?
Sorry, which were? Yeah, yeah. So, okay, I missed this. Just a second. So, this part you are asking me about, right? So, the
Yes, ma'am.
process in which, so tolerance is essentially referring to the term wherein we have the B cell not recognizing or not mounting the immune response in एक second. So tolerance is a phenomena where the immune response is not mounted against a particular antigen. Uh, Now, if this tolerance is against a self antigen, then it is known as self tolerance. However, if we have a foreign antigen, right? So maybe I am taking जैसे vaccinations होते हैं आपके, ठीक है? If in response to vaccination, yeah, if somebody is on, so vaccination is not the right example because in response to vaccination, we want heightened immune response, right? But uh, say in the situation of grafts, right? If I want the antibody to not reject a tissue graft, I want to teach this particular cell producing an antibody against this graft that you are supposed to tolerate it. So if that tolerance is ultimately acquired because of, say, certain immunosuppressants that is being given to the body or certain other drugs, right? So any sort of tolerance towards a foreign antigen is an acquired form of tolerance because it's not a natural property. We have to teach a cell to tolerate this foreign antigen. So this process, and so that is why it's acquired tolerance.
So, ma'am, in this case, we have to give a person immunosuppressant every time.
Initially, for them to not mount an immune response against it, yes. But then again, it depends from one body to another. You can have a graft getting rejected within 6 days, and you can have them rejected in 6 months or in 6 years. So, for how long a body is going to tolerate a graft is something that it, differs for every person's physiology. That's what I can think of. But so there's so tolerance and graft rejections and all those things is a little specific to individual. So we can only know that what is the phenomena that is underlying it. But for how long and all those questions, they are very specific to the medical fraternity mostly.
Okay, ma'am. And ma'am, one last thing, what do we mean
Mhm.
by signal transduction?
Signal transduction. So we'll also see signal transduction very briefly in today's class. But to answer you, second. So when I say that, okay, maybe we can look here, right? So this is the all the receptors that is present on the B cell membrane. So on the B cell membrane, what all do we have? We have this BCR or antibody very closely associated. We have Ig alpha and beta chains. And now we want that if this antigen is binding to these two antibody molecule, whatever is the so uh, this is a event. The information about this extracellular event needs to get inside the cell, right? Why so? Because whatever is the antibody production, or whether or not I have to produce more antibody or I have to differentiate into, say, a memory cell or any such information. So information center is the nucleus, right? Which is inside of your cytoplasm. So the signal which the external of this cell has received via its membrane bound receptors, this signal has to get inside the cell. Now, in itself, we do not have the C terminal region or the cytoplasmic tails of the antibody molecules. They are not very long, so they do not really get very nicely inside of the cytoplasm. So we do not have any of the signaling molecules that get a chance to bind to the cytoplasmic tails because nothing is inside of the cell. Whereas all the signaling molecules, so the signaling molecules can be certain transcription factors, they can be some kinases, certain phosphatase. So in phosphate is one of the prime molecule that plays a very important role in every sort of signaling. So you will have multiple enzymes that are either adding a phosphate or removing a phosphate, and because of that, some cascade of signals are uh, coming into picture, right? And ultimately, the information gets into the nucleus and it knows that now what do I have to make? Do I have to make gene A or B or C or whatever? So this process of your external signal coming inside the cell and then reaching the information center or the nucleus of the cell, this process is known as the process of signal transduction. Now, this not only involves your BCRs, but also the associated Ig, alpha, and beta molecules, which are very closely present along with the BCR, but they also have the privilege of having a long cytoplasmic tails. And in this cytoplasmic tails, we have something called as ITAM. So ITAM is intracellular tyrosine addition molecule, some molecule sequence, right? So there are essentially it has a lot of tyrosine residues which can be phosphorylated by some kinases. And if you see here, we have phosphorylation of tyrosine of one of the items, which is going to bring about an association of this SRC kinase by SRC molecule. And this molecule can further bind to this phosphate site because of the favorable docking position and things like that. So those are certain details into signaling pathways that is not really the concern of in immunology. So those are again something that you learn in signal cell biology while you understand about cell transduction, cell signal transduction. So cell signal transduction is a topic if you want to read further onto it. But signal transduction in the context of immunology means this, where we have this external binding of antigen. This information needs to get inside the nucleus, and the signals are transferred from the outside to inside because of certain associated co-receptor molecules. We will also see these co-receptor molecules in the T cells that we just discussed.
Okay, ma'am, got it.
Okay. Okay, so with this, we can move on to T cells, hopefully. Hopefully, you all are with me. And while we are discussing about T cells, we will try to keep our discussion as closely possible towards it being like a comparison to what we have been discussing so far, right? So just starting from the basic, we have the T cells, which are mostly effective. So B cells are producing antibodies, which can reach anywhere in the circulation, and any sort of soluble antigen or pathogen, the antibodies can clear off. But consider a situation where we have the antigen inside of the cell itself. Now, antibody is either on the membrane or it gets out into the circulation. What about this antigen? So, any sort of intracellular antigen is acted upon by T cells. So they are effective in dealing with intracellular antigens, such as maybe some intracellular parasites or intracellular viruses, or also for the cancerous cells. So cancerous cells are taken care of by antibody also, and we can also have the death of cancerous cell, which is mediated by the cytotoxic T lymphocytes. We have discussed, I hope you understand the term, where we have some CD8 positive T cells which can directly kill a cell. So that is the role of T cell, and that is the limitation of B cell that we deal with. So the difference from a B cell is that it so B cell receptors or antibodies can directly bind an antigen by recognizing the epitopes present on this antigen. But a T cell can recognize an antigen only if it is presented in association with MHC molecule. Again, we have discussed this. I'll not get into it. If any one of you fail to understand it, you can stop me, right? So I'll try to go over what and not mention again what we have already discussed. But if one of you is not able to understand, please stop me and then we can take that discussion. So it requires presentation of antigen along with the MHC molecule. And the second fact is that when they are activated, so antibodies can reach anywhere in the circulation. You can have an antibody secretion from one of the lymph nodes in, say, your arms, but those antibodies can also reach the gut, or maybe it can reach your legs. Maybe the site of infection is there. So they are long range activation, long range effector molecules. But upon activation, your T cells only act in a short range. So if you're talking in context of T helper cells, which are producing cytokines, cytokines is produced in a localized periphery. Or if you're saying that it's a cytotoxic T lymphocyte, which has to directly interact and kill a cell, again, we will have the CTL differentiation happening somewhere near the site of infection or near the site of immunogenic challenge, right? So these are short range. Why is that an important feature? Because this ensures safety of immune response. So for B cells, we had multiple checks, and eventually, even if we have, say, one antibody molecule which is, say, self-reactive, maybe it is recognizing some antigen which is expressed on one of your body cells, right? Or maybe a soluble form of protein that some malfunctional cell is giving out. So if you have your antibody molecule active against this, mounting an immune response against it, the chances of it being very fatal is less. Why so? Because firstly, self education for B cell has happened. It also happens for T cell. But the problem here is this T cell will kill your cell, or it can wrongly instruct your B cells to assume certain reactive phenotypes. So the amplification of a negative immune response is much faster if the fault is in your T cell effector response. One, it can, if say we have a self reactive T cell that is recognizing your liver cells, in no time you will have most of your liver cells being attacked by the T cells, the CTLs. If, say, you have a T helper cell which is recognizing a self antigen being presented by one of the antigen presenting cells, and because of it is producing pro-inflammatory cytokines. So now B cells, which somewhat produce certain antibodies which might or might not even get a chance to interact with some self antigen, those cells are now further stimulated to produce more antibodies. So the chances of encounter of a faulty antibody to a self antigen further is augmented because of faulty production of cytokines. So the in most of your autoimmune disorders, you will find that your the severity of consequence is much higher if the involvement is of a self-reactive T cell rather than your self-reactive B cells, right? So because so the summary being that because the consequence of self-reactive T cells amplify the negative response in a much faster, in a much heightened amplitude, we wanted to ensure the safety of immune response. So the body has smartly designed that the range of action of the T cells is shorter, much shorter compared to the range of action of your antibody molecules, which can reach anywhere in the circulation. Okay. So from the T cells, so this is a basic difference from the mechanisms of T and B cells. We have also looked into it in the very initial classes where we discussed the adaptive immune responses, right? Now we'll quickly move on to whatever is the receptors on your T cells. So first thing is the main primary T cell receptor, right? So the primary molecule that does the job of binding an antigen. Now, T cells, unlike the B cells, can only have membrane-bound molecules on its surface. So it's never going to be that you have certain TCRs that can come out in the soluble form, because the gene fragment that might be encoding this protein always have a hydrophobic domains, right? So we saw that during our principle of how an antibody can be soluble or membrane-bound. Taking from that concept, we have a lot of hydrophobic amino acids. Because of the hydrophobicity of these amino acids, there is a transmembrane domain, and we will always have T cell receptors which are membrane bound. Now, these T cells can express up to 10 to the power 5 TCR molecules per cell. So, one cell can have up to these many TCRs on its surface. And the TCR, similar to the BCR, are also associated with a signal transducing molecule called as the CD3 complex. So right there, right now we just saw, we can revisit it, that there is a signal transducing molecule, Ig alpha and beta, along with the BCR complex, which is essentially having a lot of ITAM sequences, which can be phosphorylated or dephosphorylated because of the and contribute to the signaling mechanisms. Similarly, in TCRs also, we have a signal transducing molecule, a co-receptor molecule called as the CD3 complex. But let's just go one by one. So TCR is a membrane-bound immunoglobulin, and if you look here, so most of the T cells in our body are of the make of alpha and beta type. So which means we have one alpha chain and one beta chain which is linked together using disulfide bonds. So now try and compare this structure from your, recall the structure of an antibody molecule, right? So there we had a Y-shaped structure. We had heavy chain molecules with some domains, and then okay, I hope you recall it, right? So if I just give you everything on the plate, you will not try to use your brain cells. Try and recall the structure of antibody, and you would remember that there was a certain domain, a variable chain region, or the variable domain of your heavy and light chain, and we had constant chain of your heavy and light chains. Every different domain having disulfide bond which was binding them together, and we had two polypeptides. Both the heavy polypeptides also linked to each other using disulfide bond, and we additionally had the variable and the light chains, heavy and the light chains also having disulfide attachment. So there we had four polypeptide molecules having coming into association because of the disulfide bonds, right? Here we have two such polypeptide molecules where we see that different domains in it are also having some intra polypeptide disulfide bonds, like here, and we also have some interpolypeptide disulfide bonds. And very similar to the antibody molecule, we have a variable region and a constant region in both these polypeptide molecules. So here what we see that there's one chain called the alpha chain and another chain called the beta chain. There is one variable region on both of these chains and one constant region in both of these chains. Now, if you look here, roughly, I've added some details to it, that roughly we have 60 to 75 amino acids, which is present in this hypervariable region that you see, and in the variable domain that you see. And in this variable domain, like the antibody molecule, which had certain hypervariable region, we again have some hypervariable region in the variable domains of both the chains. One interesting fact is that in the variable region of your beta chain, there is a fourth hypervariable region where we do find a lot of variability coming in. But interestingly, it is noted that this region do not contribute to the binding of antigen. So they do not classify it as a CDR. So full form of CDR say it's a complementary domining region, but this doesn't determine the complementarity of binding of this TCR to an antigen molecule. It's just there, right? So we have a fourth domain of hypervariability, which do not contribute to antigen binding, but it's just a fact that people have noted. So similar to the antibody molecule, we have three CDRs in alpha chain and three CDR in beta chain as well. Now in the constant region, so as the name suggests, the sequence of amino acid here is pretty constant. And this connecting sequence is where we have the possibility of formation of disulfide bond between these two chains. There is a transmembrane segment of 21 to 22 amino acid. And interestingly, all the residues here are mostly the positively charged amino acids, right? So most of the amino acids here are positively charged, and this we will look into it while we discuss signal transduction for T cell receptors in the next class, that this positive residue actually favors the interaction of the co-receptor molecule, the CD3 complex. And since CD3 in itself is the signal transducing molecule, so CD3 interaction to TCR is facilitated because of this positive residue, and CD3 in turn communicates the signals to the interior of the cell. Compared to the antibody molecule, the cytoplasmic tails here are still somewhat longer. So it's roughly 5 to 12 amino acid, but still not long enough for it to help in signal transduction. Okay, so that is how the structure of a TCR looks, where it's a heterodimer of alpha and beta chain. But if you remember from again from a very previous class of us, where we were discussing about the tertiary lymphoid organ, where there was we were discussing about the cutaneous associated lymphoid tissue, the CALT. We saw that we had certain Langerhans cells and some del TCRs which were gamma del positive, right? So therein the point is we do have very rare population of T cells wherein the TCR is of the gamma del type. So instead of this being an alpha chain, it is a gamma chain, and instead of this being a beta chain, it is a del chain. Obviously, the number of amino acid corresponding to it, if it's a del or a gamma chain, it is going to vary. But if you just have a structural overlook at it, it looks like the same, wherein we will also have certain variable region and some constant region, right? But it's just for you to know the T cells mostly are having alpha beta TCR, but we have some small population of T cell which have gamma del uh, type of TCR. They are low in number, but their numbers increase in response to an infection. So if there is an infection of, say, a bacteria or something, their numbers, the number of T cells where gamma del receptor TCR is present, their numbers have been found to be increased. They do not require antigen presentation and processing. So alpha beta T cells, TCRs, it is so MHC restriction, antigen being presented along with the MHC and the T cell only recognizing that antigen if it is bound to an MHC. All of those facts they hold true if the TCR is of alpha beta type. If it's of gamma del type, we do not such TCRs do not rely on antigen presentation. They can in itself bind to the antigen directly. It has been found that the gamma del TCRs are also present on some cells of the innate system. As I was just saying that the scar tissue, those were the features while we were discussing about the barriers in innate system. So they may be considered as a part of, as an arm of innate immune response, right? And all the so your alpha beta TCR along with it, you can have either a CD4 positive or a CD8 molecule present with it. We'll just come to CD4 and CD8. We'll have a look at their structures. But all the TCRs of gamma del type are only associated with a CD8 plus cells. And it has been shown that they can function in recognizing the phospholipids and glycolipids. So just a quick comparison of your alpha, beta, and gamma del TCR. So your along with your alpha beta TCR, you can either have CD4 plus or CD8 plus molecules present, but along with gamma del TCR, you will only and only have CD8 plus molecules present. MHC restricted होते हैं alpha beta TCR. There is no MHC restriction of your gamma del TCRs. Antigen that the alpha beta TCR can recognize is always and always a peptide because it's only the proteinaceous antigens that get a chance to get processed and get associated with the MHC molecules, and alpha beta TCR is a MHC restricted receptor. But over here, the antigen that the gamma del receptors bind to is a phospholipid or a glycolipid antigen. Okay, so this brings us quickly to the T cell co-receptors that we were just discussing, which can we can either have a CD4 or a CD8 co-receptor molecule associated with a TCR. So if we look at the CD4 molecule, it's a monomeric unit. So I think so far this is the first monomeric unit that we have encountered, apart from certain adhesion molecules that we have discussed while we were discussing the cell receptors. Anyway, this is a 55 kilodalton monomeric unit, which further has 4 domains. All the domains are linked by disulfide bonds. There is a transmembrane domain and a intracytoplasmic domain. Compared to this, in the CD8 molecule, we again have a heterodimer where we have one alpha beta associated by disulfide bond with just one domain. Or it is also a possibility that instead of alpha beta make, we can have a alpha alpha homodimer of CD8 molecules, right? So CD8 molecule can be formed by this alpha beta chain, or it can also be formed by the alpha alpha chain. In that case, it's a homodimer. And each case, we have both the chains having one extracellular domains. The function of these domains are essentially to be bound by some receptor or the other so that the signaling can be facilitated. They can only recognize antigens present with class 1 MHC molecule, whereas for CD4, we have them to be class 2 MHC restricted. Okay, so along with the association of TCR with the core receptor, we can have TCR. So in general, the binding of TCR with MHC with antigen is a weak association. So the binding of antigen and antibody is with a lot of energy release. That's why the binding is of a much higher affinity, but the interaction of your TCR and the MHC antigen complex is weaker. So we do it is very, very required that as the interaction of TCR and MHC molecules begin, there is a overexpression of cell adhesion molecules. So we initially have some adhesion molecules already in place, but as soon as the cell tries to interact with the prospective antigen MHC complex, the number of the expression of these cell adhesion molecules is upregulated. Now, which molecules are we talking about? We're talking about CD2, LFA, CD40L. So different names, different, each of them having different functions. And if you look here, you will you will understand that one molecule is essentially not just performing one function. So if you look here, CD4 molecule is also helping in binding to the ligand, the class 2 MHC molecule, which is a very important phenomena for your signal transduction. But apart from that, it is also helping in adhesion. So it helps the two cells to come together in a coherence. Similarly, class CD8 molecules are important for recognition to the class 1 MHC molecule, but also helps in adhesion. CD2, or I think LFA stands for leukocyte factor adhesion. You can have a look at it. So CD2 along with LFA, it binds to CD58 and LFA3 on your antigen presenting cells. The primary function of this is adhesion, but it can also have a role in signal transduction. You can just go through this table. So CD28, and another very important molecule, CD28 interacts with B7 of your antigen presenting cells, and they do not have any role to play in adhesion. So B7 is a very important ligand molecule expressed on every antigen presenting cell. So along with class 2 MHC molecule, uh, for a cell to be an antigen presenting cell, it should have the ability of producing B7 on its cell surface. So B7 binding to these two receptors on your T cell helps in the process of signal transduction. Similarly, CD45 and CD45, which are these corresponding ligands, will be present on the cell that it either has to kill or a antigen presenting cell, and we can have that having a role in adhesion or in signal transduction or both of them. So just some details. I don't think anything in that detail is going to come up, but for your information, you can always try and memorize these things. Okay, so finally, the T cell receptor complex or the TCR CD3 complex. Now, CD3 is so the this entire CD3 complex, as the name suggests, it's not one protein that is expressed on the surface. It's a complex of multiple proteins, right? So this can be formed again. So this gamma, eta, del. So these are all Greek letters, and instead of like calling it a AB chain, or they have called it alpha beta chain, instead of calling it a Z and E chain. So it's just different polypeptides that have been given different names. So instead of giving it a complete name, they have given it A, B, C, D, such sort of numberings or namings, right? So which two chain of immunoglobulin? So we have, so this entire thing that you see here is a CD3 molecule. So this entire CD3 molecule is constituted by a gamma epsilon association, right? So we have one here which is of gamma epsilon type. We can also, we could have also had it of, so we also have it of epsilon and del type. So if you look here, the structures look almost the same. The variance only comes from the fact of the number of amino acids present in total, and all of this, so this ITAM sequence. So the things that you see in blue are the ITAM sequence that we have been seeing, that they have a role in signal transduction. And it can also be of zeta and eta type, or zeta and zeta type. So here is a TCR. Along with this TCR, we will have a CD3 complex. CD3 complex, this complex itself is either constituted by, it's constituted by all of these polypeptides in the present in near vicinity: gamma epsilon, epsilon del, zeta zeta, zeta, and or it can also be zeta and eta type, right? So instead of this be in some other T cell, instead of the CD3 molecule having zeta and zeta type, it could have been a zeta and eta type of chain. You have to only remember that in gamma and epsilon component dimer of CD3, we have one ITAM motif on either of these chains. So in total, the dimer has two. In epsilon del type also, we have one ITAM sequence on either chain. But in this dimer of zeta zeta or zeta and eta, we have three ITAM sequences on each of the chain. And if it's a dimer functioning together, in total we have six. So CD3, thankfully, it's an invariant. So it's that same on all the chains. So if in a particular human you have a CD3 of zeta and zeta type, all the cells will be of zeta and zeta type. In some other individual, it can be of zeta and eta type, right? So CD3 is invariant. It's the same on all cells. And CD3 is also an exclusive marker for T cells. So for sometimes, because of scientific interest, we want to isolate or mark a certain cell population. So CD3 is the most reliable marker for T cells, and many times we have anti-CD3 antibodies for tagging our T cells. So it's an exclusive marker for T cells and it's invariant. So whatever is your uh, combination of CD3 on one cell is going to be the same on all the cells. The function of it is because it is having an ITAM motif, so it's immunoreceptor tyrosine based activation motif. So that's the full form of ITAM. So it's an immunoreceptor tyrosine based, which means that there's going to be certain tyrosine residues in this sequence, and tyrosine kinase can act on these tyrosine residues and it can add a phosphate group. Because of the addition of phosphate groups, there can be some changes in the overall inside of the cell, or we can have certain signaling molecules that can now come and bind to it, as we just saw for B cells. And those molecules can reach from one part in the cell to the other part in the cell, get into the nucleus, and do all the signal transduction that ultimately needs to result. Right. So apart from CD3 being a signal transducing molecule serving as an exclusive marker for T cell, they also act as chaperones. So if you have some background of cell biology, you would know that chaperones help in proper folding of the protein. So they also have act as chaperones that helps in transporting your TCRs to the cell surface. So while we have a TCR in association with the CD3 complex, it's the signals that are present from the CD3 molecule that says that this entire complex has to get expressed on the cell surface and do not have to go anywhere out of the cell. Right. So again, so this is What we have a TCR. Along with that, we have four receptors, which can either be CD4 or can be CD8. But in both CD4 and CD8, we have a complex, a CD3 complex, which helps in transducing of signal. Okay, so this brings us to just a nice diagram that can help us visualize all the interactions that we just dealt with theoretically. So if you look here, there is a T helper cell. Just have a look. I'll punish. Okay. So nothing much to explain here. It's just the same thing. So here we have the antigen. If it would have been extracellular, the antigen presenting cells have taken it in. They have processed the peptide, and this peptide, in association with class 2 MHC molecule, is now being presented to the TCR in its TCR CD3 complex. So these are all CD3 molecules that you see here. Alongside, the CD22 molecule present on antigen presenting cell is interacting with CD45R. So this association is helping in adhesion. Similarly, B7 and CD28, which helps in adhesion as well as is a co-stimulation for this binding. So for this particular signaling. LFA and ICAM interaction helps in also cell adhesion, and I think CD2 and LFA3 helps in both cell adhesion as well as signal transduction. Similarly, if you look here for TC cells, the antigen in target cell is already inside the cell and it is getting presented along with class 1 MHC molecule after processing. This is recognized. This over here you see is the CD4 molecule, which is associating with one of the domains on your class 2 MHC molecule. Here, it's this dimer, right? So we have a heterodimer or CD8 molecule, which is recognizing a particular domain on your class 1 MHC molecule that is presenting this antigen to your TCR CD3 complex. And eventually these associations are also present. And whatever is the signal now that the CD3 is going to release or the binding is going to produce, it is accordingly bringing about a change in your gene transcription and translation. Okay, so this finishes with all the receptors that are decorated on the surface of T cells. And now if you get in this, so this is more so like an so what we have already discussed for B cell, it's exactly the same. Some diagrams I have kept to discuss. So just go through it once again so that there's no confusion while you're trying to read it on your own. So just like the organization of your BCR, so we had multi-gene arrangement and the possibility of rearranging these genes to bring different combinations together. And similarly, the same thing is happening. The organization is similar, multi-gene arrangement, and different gene segments can be brought about together by recombination, wherein we have the same enzymes, the RAG enzymes and RSS sequences, the two-turn, three-turn motifs, heptamer, nonomer, everything remains the same, right? The entire machinery is same. So, what happens? Like in the light chain, we were having VJ rearrangement, whereas for the heavy chain, we were having VDJ rearrangement. Here, if you look for TCR, if it's a alpha beta TCR, alpha chain is undergoing VJ recombination and VDJ recombination is being undergone by your beta chain. For a gamma del TCR, your gamma chain undergoes VJ rearrangement and del chain undergoes BDJ rearrangement, right? Okay. And one interesting fact remains that in the organization of these gene segments, we have the alpha and the del regions in successivity, right? So I'll just bring it upon here. But if there is a productive rearrangement of alpha gene, it happens in a way that the constant region of a del for corresponding to a del chain is deleted because it is spliced off, right? So that we only have alpha beta TCR, which is never expressed along with a gamma del TCR. So we saw that, so this, where alpha beta and where gamma del TCR rearrangement and everything is happening, when we just look into the development of T cell, you will understand this better. But a fact remains that whenever there is a rearrangement of alpha chain happening, the C of the del chain, the constant gene segment of the del chain, is spliced off. So we will never have the expression of alpha beta TCR and the expression of gamma del TCR on the same chain. Another fact remains that the number of V and J and D segments, which is corresponding for a TCR, is less than the number of V, D, J, and J segments corresponding. So, which what this means is that, say, if the number of V gene segments correspond that is possible for a kappa light chain, say it was 200, right? Maybe I'm just quoting random figures. We discussed that was exactly the numbers. Maybe 200 V gene segments are present on the kappa locus, right? But only one of them is brought about in this joining of these J segments. But over here in alpha chain, the number is not 200, the number is say 150. So the number of such V, J and possible combinations that you will see we have. So just look here. So this compares the germline DNA, and this shows that the possible number of H chain, the present number of H chain V regions is 134, D regions is 13, J is 14. Similarly, for kappa chain, it's 85. There's no D region in light chain and 4 of the J segments. If you look here, ultimately, we also discussed this previously that the possible number of combinations, which is accounting for the diversity index of antibody molecule, comes from the multiplication of all these and all these together, right? Because any number of any combination is possible. So the total possible combination is what comes as a product of the multiplication of all these numbers together. But for TCRs, if you look now, the number of V segments is comparatively much less, is lesser for your alpha chain. So if you just make it a total, that this is somewhere around 125 V segments in total, and this goes beyond 200 V segments for your heavy and light chains together. So the idea is the number of this V, D, and J gene segments for your TCR and either alpha or gamma del. So gamma del is even more less, right? So for your because of this being lesser, the possible number of combinations that these can associate into is lesser, because obviously the numbers are small. The product of multiplication of this number is also going to be small. So this is one prime reason why the diversity account of your TCR is lesser, right? And another thing that we will also see is that there is no somatic hypermutation that is happening in TCRs. So in BCRs, the diversification was coming from this combinatorial association, the multi-gene arrangement, but it was significantly enhanced because of somatic hypermutation. Just a minute. Mm, Okay. So I think what we have we were trying to discuss is clear that the diversity index is less because one, there is the number of gene segments for your V, G, and D regions is lesser while we are talking about the TCR chains, and also that there is no somatic hypermutation that happens in your TCR. Now, okay, now focus here. So this is talking about mouse TCR. Are you encountering a lag? Okay. So this is discussing a case of a mouse TCR where we have 100 possible V alpha regions and we have somewhere around 10 possible V del regions. Now, if it's the gene rearrangement is happening for the alpha region, if you look here carefully, the del regions are present on the same chromosome as the alpha regions, right? So the del regions, if the alpha, say this V region of alpha is selected in a recombination event, whatever lies in between is going to be completely looped off or spliced off, because ultimately we will have the V region of alpha joining to the J regions of alpha only. So this entire intervening sequence, which is corresponding to the del chain, is completely spliced off and lost. And once something is spliced off and lost, the same cell can never get back this gene segment, right? So this is one way of ensuring, as we were discussing here, that alpha, beta, and gamma del TCRs are never expressed together on the same cell. Another fact that we will see is that this is an exception to allelic exclusion. So the alpha chain of TCR is an exception to allelic exclusion. What does that mean? That it has been found that when TCRs have been sequenced, the beta chain of so we are talking in terms of clonally selected population of T cell. So maybe a T cell named A. A T cell named A give rise to, because of clonal proliferation, it is giving rise to daughter cells which are A1, A2, A3, A4, A5. All the A1, A2, A3, A4, 5 cells are having the same sequence of beta chain. But the sequence, the VDJ combination of the alpha chains present in association with these beta chains have been found to be slightly different. But while we are talking in terms of specificity, you understand that even slight differences can change the specificity of a TCR molecule, right? But people have found that there remains a difference in the sequence of uh, VDJ, but the specificity of antigen is still maintained. How is that happening? This is not known, right? So we still have a lot of things uh, which contradict to whatever theory that applies in general to a number of places. But so that is what keeps science going, that we have some exceptions to the generalized principle. And one such exception is that alpha chains are an exception to allelic exclusion. So people have tried to see that because A1, A2, A3, all of them are having some subtle differences in in their VDJ combination. So maybe that we have some other allele from the other parent which is coming up and is getting a chance of getting rearranged and get expressed with the beta chain, which is same in all the A1, A2, A3 cells. But it might be a alpha chain is coming from the other copy of allele from the other parent compared to what was originally present in your this A cell that was there, right? So that is so I am just trying to give you certain facts that are true to TCRs for today's class, because the principles and all those things we have already discussed in our previous classes, right? Now for the beta chain in mouse, it is on chromosome 6. Okay, in mouse it's in chromosome 14. For humans, I think it's 9, but have a look at it. You can just find it in any textbook. You find it in QB itself. And we also have the gamma chain, and which is on chromosome 13. Now, if say because del will, if we do not have the del here, even if by chance we have the gamma chain coming into expression, it will not have something it can pair with. And so that combination is ruled out in that process. And if you look here, so again, chromosomal location and the number of possible V, D, G, and J segments, we have also looked at in while we're trying to see for the diversity index. This shows the rearrange. So here we were looking at the overall organization of the alpha, beta, gamma, and del chains. And this shows the rearranged alpha and beta chains, right? So what is present in your encoded domain is this leader sequence, as we've also previously discussed, that leader sequence is what helps in signaling of your molecule to remain transmembrane. Eventually, it is cleaved off in post-translational modification, and that's why it's not the part of your TCR. In BCR also it's the same. We have a leader sequence, but it never expresses on your antibody molecule because it's cleaved off when the protein is undergoing further modification, but it remains in the primary transcript of mRNA that is formed. This constitutes the variable domain, the V and J in alpha chain. In the beta chain, it's V, D, J. There are CDRs. Why are CDRs being formed? Any one of you, if you have gone through the previous lectures. So CDRs is essentially the point where we have the joinings, right? So this joining, because it's of the V and J, CDR3 is at the joining of V and J. This is the most hypervariable region. And what is leading to this hypervariability? It is because of the P and N addition. We discussed that while RAG is coming into picture, it is cleaving, it is forming a hairpin. The hairpin can be cleaved at any point by an endonuclease, and we can have random nucleotide addition. So these nucleotide additions can either be of the P type or it can be of N type. So P and N addition at the juncture of V and D, D and J, they are causing the hypervariable sequences, which is also CDRs, because they determine the complementarity of binding of your TCR to an antigen MHC complex. And in BCR, it was determining the binding of antibody and antigen. Now we will have the constant regions that have also come together. We will have the hydrophobic sequences, the transmembrane sequences, and the C terminal or the cytoplasmic tail domains that have all come together now from this organization of gene because of the process of VJ and VDJ recombination. There is no somatic hypermutation that happens in TCR. Again, uh, okay. So again, it's a comparison of what happens in your TCRs and immunoglobulins. So VDJ happens for your beta and del chain. Here VDJ happens for heavy chain. VJ happens for alpha or the gamma chain. VJ happens for the light chain. Alternative joining of these segments. Why was that happening? Because we had the RSS sequences where only a one turn and a two turn sequence can come together in a recombination event. So alternate joining sequences happens in this way where we have VDEL. So this is just some fact that first we have this, and then this coming together. Here we simply have the VDJ joinings, right? Junctional flexibility, which allows for random nucleotide insertion, N region nucleotide insertion. Just everything that we have already discussed. It's just a comparison for T cell and your immunoglobulins. If there's anything that is not clear, you can stop me in between. We'll quickly complete comparative account of the entire thing, and then we can stop prior to the development of T cells. Okay, a quick overview. So alpha beta TCR, they have compared it with the immunoglobulins, the membrane-bound immunoglobulins. Here the components remain alpha beta. Here it's the heavy and light chain, the number of V and C domains. So there's one V and one constant domain, one variable and one constant domain on each chain. On heavy chain in immunoglobulins, we have one variable and 3 to 4 constant domains, and on light chain, one variable and one constant domain. So valency means that how many antigens can a particular receptor bind to. So valency of a TCR is 1 because there is just one binding cleft. Once this is alpha and this is beta, and antigen is bound over here. So the valency is monovalent. However, for your antibody, which is this Y-shaped molecule, we had the antigen possibly binding on either of these ends. So it's a bivalent molecule. Next, which is the nature of antigen, the TCRs can only recognize protein antigens, whereas the BCRs, okay, whereas the BCRs, they can also recognize carbohydrates, lipids, and along with the protein molecules. Now, antigen recognition is for a small linear sequence of peptide which is presented to it with a MHC complex in a MHC complex. However, antibody recognizes epitope. So epitope can be from, okay, so this is one thing that we have not discussed so far because we quickly skipped through the discussion of antigen antibody interaction. Those are very straightforward things. I'm sure you have come across it. So while it's a processing happening, so imagine it's a, so let's just say that the entire antigen is like a complete ball of, say, wool, right? You all have seen like a tangled mass of wool. In this wool, because of this folding, we have, say, this cleft forming, right? This small cleft forming, which is recognized as an antigen, and it can be recognized by a, yes, sorry. So, so idea is that the folding of the molecule has formed a particular structural feature, which is the epitope of an antigen, and it is getting recognized by the antibody molecule. But when processing of this peptide, so now let's just say that the same uh, ball of wool has got inside the cell, right? So inside the cell, it is going to be subjected to a lot of processing. In the process of processing, we have this spool of wound opened up, and then it is going to be cleaved off in small fragments, and these fragments are going to be presented as a linear stretch of peptide along with the MHC molecule. So the one possibility there remains that while this is getting processed, so this small region, which was an epitope of resulting from entire folding, we have a small part of it remaining here and a small part of it remaining here. So now if you give this process peptide, now if you give this process peptide for to this particular antibody, it will not be able to recognize it because overall folding has been linearized, and because of this particular epitope organization can no longer be recognized by the same antibody molecule. Okay. But the feature of your T cell is that maybe because of this linearization, we have this section, okay, wrong color. What do I use? Okay, is all. Maybe this section now is when this small part is presented along with a MHC molecule. This can be recognized by the PCR. Okay. So this is what antigen recognition here speaks for, that antigen recognition is against a small linear sequence of peptide. What linearized your entire protein is the processing part of antigen, whereas the antibodies can recognize antigens which are in variety of structures and shapes. Okay. The affinity for antigen is comparatively less, and that's why we saw that there's a lot of cell addition molecules that have to come into the picture, whereas the affinity of binding of an antibody to antigen is very high. There is nothing like isotype switching because it's not like a alpha beta switches to a gamma del or anything like that. Nothing like that happens. No isotype switching. But for antibody, we saw that there's a lot of isotype switching, and we also understood that what brings about that those switchings, right? There is no secretory form of PCR, whereas after the antigen binds to the membrane bound immunoglobulin molecule, it is possible that the B cell differentiates into a plasma cell, and the primary function of plasma cell is to produce antibody in secreted forms. Okay. Okay, now just a small concept. I don't think we will be able to get into this today. I just thought I'll bring a brief overview, but then we'll put everything to the next class. Not a problem. So the concept of self-MHC restriction of PCR. So we have so far we'll quickly try and I don't know what's wrong with the net today. Okay. So self-MHC restriction. So we have been saying so far that the TCR can only recognize antigen if it is presented along with the MHC molecule. But the MHC molecule of one's own body, right? It is not like that if my cells in my body, the MHC molecules of my body is presented with an antigen, and maybe someone else is presenting the same antigen to the T cells of my body along with their MHC molecule, this particular T cell will no longer recognize the antigen, same antigen complex with MHC of a foreign individual. So this phenomena where the TCRs can only recognize self MHC molecule and antigens presented. Okay. So this phenomena was decoded by two individuals, Doherty and Zinker-Nagel, and they also won a Nobel Prize in 1996 for this same purpose. What did they do? So they simply did a experiment wherein they say that so this here you see H2 to the power K, it's cell line, right? But it's a strain of the model that they have used. Okay. So maybe this one individual that they have used, they have named it as H2K. So it's a strain. So they isolated CTLs, or what are CTLs? The CD8 positive T cells. They isolated CTLs from this individual, and they saw that when we have, say, some target cell in the same individual, we have a viral infection in this individual. This virus is residing inside a cell, which the CTL has to now go and kill. Now, the virus, when it is present in the cell of the own individual, it is expressing self-MHC molecule. The viral peptides are going to be processed, presented with the self-MHC molecule, and the same individual CTLs can very well recognize this peptide MHC complex and ultimately bring about killing function. The other possibility is that, so even TCRs are specific to, they are committed to what kind of antigen and MHC molecule they can recognize. This same CTL, if they now subject these cells in the same individual to a different virus, and the viral protein presented in a processed form with the same self-MHC molecule to the same CTL as was here. But since the processing now is different, right? It can also be that it's the same virus, but since processing of a peptide can be in different. So if you see here, the cut, it can also be this peptide fragment gets presented. There's also a possibility that this peptide fragment gets presented. So while a protein is processed, all the different peptides component can be presented with to the TCRs with the same MHC molecule. It's only the TCR of a particular specificity that can recognize a particular peptide, right? If from the same virus, a different polypeptide is presented with the self-MHC molecule, this CTL can no longer kill this cell. Why so? Because here the antigen itself has changed. The specificity of this CTL was against the viral peptide A. So it cannot bind to viral peptide B anymore, despite the fact itself. We understand this so far. But the interesting fact that was noted was that there is also no killing. If instead of H2K, they have a cell of a different individual presenting the viral peptide A. Why so? Because here the CTL is from a different strain, the target cell is of a different cell, is of a different strain. So, despite the peptide specificity maintained, we cannot have any killing function noted in this target cell because it's a non-self MHC molecule. So, this is the fact this is what is known as self MHC restriction of T cells. And where is that these T cells are learning about it? It is happening inside the thymus. So while we discuss this, we'll try and understand this. For now, you should know that while the TCRs are developing or while the B cells are developing inside the thymic epithelial cells, which are purposefully presenting all the antigens and different components to the developing T cells, that there happens the recognition of cell. So whichever MHC the developing T cell has encountered in the thymus, it recognizes antigens complex only with those MHC molecules. So whatever is the MHC molecule present on your thymic epithelial cells, the T cells are getting an education that these are my self MHC molecule, and only when the antigen is presented along with the self MHC molecule, I can bind to this complex and I can mount an immune response, right? So that's it. We will stop here for today, and for the next class while we discuss T cell, मैं तो बेटा नहीं check करूंगी. So paper and everything is going to be checked by your different faculties. I don't know where the NPTEL sends the exam answer sheets or anything like that. Right? So let's just focus on understanding so that whosoever checks the paper, we do not have a problem. If there's any doubt that I can help you with, any understanding that you want me to revisit, we can start taking that up. Okay. I am not involved anywhere in the examination process. I am only here to help you further and better understand the topics for immunology, जिसके recorded lectures आप लोगों को मिलते हैं. Okay, we'll wait for another couple of minutes. If any one of you wants to ask anything, you can unmute yourself. I am right around, or else we'll end the session. Bilingual, if अगर आपको मतलब if you are not comfortable, अगर इतना comfortable नहीं हूँ आप इंग्लिश में, तो अगर कोई कॉन्सेप्ट आप चाहते हो कि मैं आपको हिंदी में रिविजिट कराओ तो यू कैन टेल मी द नेम ऑफ दैट कॉन्सेप्ट। अदरवाइज अगर आपको कंफर्टेबल नहीं था लैंग्वेज में, आप मुझे क्लास के शुरू में बोलते, सो आई वुड हैव ट्राइड कि मैं बाइलिंगुअल मोड में क्लास लूँ। बट जो भी आपको रिकॉर्डिंग्स अवेलेबल होंगी और वो उसी लैंग्वेज में होगी जिस लैंग्वेज में मैं अब तक बात करती आई हूँ।
Hello, good evening, ma'am.
गुड इवनिंग।
And this is Rajkumar.
या आई से योर नेम, प्लीज गो।
Uh, I was asking कि ये जो चैप्टर्स हैं, लेक्चर्स हैं, बाइलिंगुअल के लिए, जो कि
मैं
वैसे प्रॉब्लम नहीं है, जस्ट बिकॉज ऑफ द कंसेप्ट क्लैरिटी आई नीड इट
हम्म, नहीं,
इन द
आई
बाइलिंग.
अंडरस्टैंड योर कंसर्न, बट मैं यही कह रही थी कि अगर किसी भी कंसेप्ट में आप चाहते हो कि मैं आपको एक बार फिर से बताऊं, मैं आपको हिंदी में समझाऊं ताकि आप बेहतर समझ पाओ, तो आप मुझे बता सकते हो। मैं उसको वापस आपको अभी समझा सकती हूँ। बट आपके पास यही रिकॉर्डिंग्स अवेलेबल होंगे अगर हम जब यूट्यूब पे डालेंगे, या जो भी आपका रिकॉर्डेड लेक्चर्स भी है वो सब आपको इंग्लिश में ही मिलेंगे, मिले हुए हैं, राइट? तो आई डोंट थिंक कि उस पे कोई भी हिंदी का ट्रांसलेशन या समथिंग लाइक दैट एटलीस्ट ऑफ लाइव सेशन का तो आप लोग को नहीं मिलेगा। क्योंकि जो भी हम अभी बात करते हैं, जिस भी लैंग्वेज में बात करते हैं, वही रिकॉर्डिंग्स आपको एज इट इज मिलेगी। तो अगर आप चाहते हो कि आपको कुछ समझ में नहीं आया है और मैं आपको उसको आपको हिंदी में समझाओ ताकि आप बेहतर समझ पाओ अभी या आगे की क्लास में या कभी भी, तो आप मुझे अभी बोल सकते हो, या जब भी आपको समझने में दिक्कत हो रही है आप मुझे तब बोल सकते हो। बट एनी सॉर्ट ऑफ ट्रांसलेशन ऐसा कुछ आप लोगों को अवेलेबल नहीं किया जाएगा, क्योंकि ऐसा कुछ नहीं हो रहा है।
ओके, नेक्स्ट टाइम से जब भी ये लैंग्वेज.
श्योर श्योर श्योर। इनफैक्ट नेक्स्ट टाइम से अगर आप अभी अगर रिकॉर्डिंग्स को रिविजिट करो, आपको ये रिकॉर्डिंग्स मिलेंगी। आप नेक्स्ट क्लास में, अभी की क्लास है इससे पुरानी कोई भी क्लास, देखो, दीज क्लासेस आर अरेंज्ड बाय एनपीटीईएल। ये आप लोगों की डाउट सॉल्विंग क्लासेस है। बट एक जनरल ट्रेंड जो हम लोगों ने ये पाया है कि आप लोग डाउट्स लेके नहीं आते हो, एंड हम लोगों को क्लास कंडक्ट करनी है तो हम लोग इसको थ्योरी क्लास की तरह कंडक्ट करते हैं, ताकि अगर आपके कोई डाउट्स है भी जिसको शायद आपको पूछना नहीं आ रहा है या आप कॉम्प्रिहेंड नहीं कर पा रहे हो, आप नहीं समझ आ रहा क्या पूछना है नहीं पूछना है, तो आप इस क्लास को अटेंड करो। ठीक है? बट अगर हम आगे तभी बढ़ेंगे जब आपके पुराने सारे डाउट्स क्लियर होंगे। क्योंकि कोई प्राइमरीली कोई सिलेबस नहीं है जो मुझे पूरा करना है या कराना है। क्योंकि आपको रिकॉर्डिंग्स पूरे मिलेंगे और क्वेश्चंस आंसर्स एग्जाम्स असाइनमेंट सब आपको उसी रिकॉर्डिंग्स के हिसाब से मिलेगा। हमारा क्लास लेने का पर्पस डाउट सॉल्विंग क्लास है। तो आप लोगों को जब मेल्स भी जाते हैं, आपको डाउट सॉल्विंग सेशंस की तरह मेल जाते हैं और लाइव इंटरेक्शन के नाम पे मेल जाते हैं, राइट? तो अगर आपको, इसलिए हमें कोई जल्दी नहीं है कि हमें इतना खत्म करना ही है। तो जो इम्पोर्टेंट कंसेप्ट्स है या जो थोड़े समझने में मुश्किल कंसेप्ट्स है, हम उन कंसेप्ट्स को अच्छे से डिटेल्स में कवर करने की कोशिश कर रहे हैं। अगर पुराने लेक्चर्स में आपको कोई भी प्रॉब्लम है, जो कि रिकॉर्डिंग्स आप पे अवेलेबल है, अगर आपको लग रहा है कि इसको मैं बेहतर समझना चाहता हूँ, तो हम नेक्स्ट क्लास की शुरुआत उन्हीं डाउट्स को आपके लिए जो भी भाषा कंफर्टेबल है हम उसमें बात करने की कोशिश कर सकते हैं। ठीक है? तो आगे भी आप मुझे रोक सकते हो, और अगर पुराने किसी लेक्चर में आपको दिक्कत है तो आप वो भी पूछ सकते हो। ओके, सो इट्स दैट इज ऑल फॉर टुडे। आई कैन क्लोज द सेशन।
Ma'am, one last thing I want to ask.
गो अहेड।
Ma'am, there is written something above CTL, that is H hyphen 2 raised to the power K. So what
Mm-hmm.
does it refer and what does it
It's
mean
just a
actually?
strain of, so this is an experiment. So if you just have a look here, it's the ligand, if you, the figure ligand, if you see that a particular TCR is specific to only antigenic peptide and self-amino in this example. So it's an example where a particular host of this strain, so जैसे आप जीनस और स्पीशीज और ये सब पढ़ते हैं, तो जीनस स्पीशीज के बाद भी स्पीशीज को हम डिफरेंट डिफरेंट स्ट्रेन्स में क्लासिफाई करते हैं फॉर एक्सपेरिमेंटल पर्पस. So just assume it like a name for a particular individual. So it can also be like हमने राज का सीटीएल लिया और राज का टारगेट सेल के साथ हम एक्सपेरिमेंट कर रहे हैं, राइट. इट कैन बी, it's just a name for the host from which we have taken a CTL or the target cell we are discussing for anything like that. It's not relevant, it's not something relevant to concepts in immunology.
So, ma'am, it is the name of the host from which we have taken that CTL.
Yes.
Okay, ma'am. So, ma'am, that we are using in lab and we have designed the targeted cells with respect to that cell only that we have isolated.
See how the experiment is carried on. Let's say I have a mice, okay? The mice strain or the name of the mice is H-2K, right? इस माइस को इस माइस से हम लोग सीटीएल्स आइसोलेट कर सकते. माइस को हमने पहले वायरल इन्फेक्शन दिया और इस माइस से, क्योंकि इन्फेक्शन होगा, वायरल इन्फेक्शन होगा, तो इंट्रासेलुलर एंटीजन प्रेजेंट है, तो सीटीएल एक्टिवेशन होगा. वी कैन आइसोलेट सेल्स फ्रॉम द बॉडी ऑफ द माइस सर्कुलेशन से या साइट ऑफ इन्फेक्शन से, और हम मार्कर के बेसिस पे टी सेल्स को अलग से आइसोलेट कर सकते हैं. फिर उनको CD8 या CD4 के मार्कर के बेसिस पे हम उसको CTLs या टी हेल्पर सेल्स की तरह भी क्लासिफाई कर सकते हैं. सो दिस इज हाउ एन एक्सपेरिमेंट इज परफॉर्म, कि हमने एक माइस लिया, उसको वायरस इन्फेक्शन दिया, इस माइस के बॉडी से हमने CTLs को आइसोलेट कर लिया, ठीक है. आइसोलेट CTLs को भी हमने किया, और हमने जिस भी सेल में हमने वायरस का इन्फेक्शन टारगेट किया है, मानो हमने एपिथेलियल सेल्स के अंदर ऐसा वायरस चूज किया जो एपिथेलियल सेल्स के अंदर अपना घर बनाता है. तो जैसे हमने एपिथेलियल सेल्स को अलग किया इस माइस से और सीटीएल्स को भी अलग किया, और इन दोनों को आपस में हमने सेल कल्चर मीडिया में इंटरैक्ट करने दिया, हमें दिखा कि जो एपिथेलियल सेल्स है माइस की, इन वीवो कंडीशन, लैब कंडीशंस में, बॉडी के बाहर की इन विट्रो कंडीशंस में हमें दिखा कि एपिथेलियल सेल्स का नंबर कम हो गया, मतलब सेल डेथ हो गया. सिमिलर एक्सपेरिमेंट हमने एक एपिथेलियल सेल, एक ही टाइप के एपिथेलियल सेल का नाम लेके से हमने आ, कोई दूसरा एपिथेलियल सेल लिया जिसका हमने देखा कि उसमें सेल डेथ नहीं हो रहा है. तो जब उस एपिथेलियल सेल को प्यूरिफाई किया गया तो पता लगा कि जो पेप्टाइड प्रेजेंट हो रहा है वो अलग है, और जिन सेल्स में डेथ हो रही है वहां पे वायरल पेप्टाइड कुछ अलग है. दैट वी कैन अंडरस्टैंड, क्योंकि एक प्रोटीन को जब एक ही सेल ने प्रोसेस, अलग अलग सेल्स ने प्रोसेस किया, तो हो सकता है अलग अलग वायरल पेप्टाइड के साथ MHC का कॉम्प्लेक्स बना, और CTL की, पर TCR की तो स्पेसिफिसिटी सेम है, तो वो किसी एक ही टाइप के पेप्टाइड को रिकॉग्नाइज कर पाएगा. बट इंटरेस्टिंग पार्ट, MHC रिस्ट्रिक्शन की पार्ट को हम कहाँ से समझ सकते हैं, कि जब यही वायरल पेप्टाइड A किसी अलग स्ट्रेन के, तो H-2D स्ट्रेन के एपिथेलियल सेल्स को भी हमने इसी वायरस से इन्फेक्शन दिया और उसको आइसोलेट किया, और हमने पाया कि इस सेल पे पेप्टाइड A का एक्सप्रेशन है. अब जिस CTL से फर्स्ट केस में जहां पे आपका H2K का एपिथेलियल सेल जो डेथ हो रहा था, वहां जो CTL था, उसी CTL को उठा के जब हमने इस कंटेक्स्ट में इंक्यूबेट किया, तो यहाँ हमें दूसरे स्ट्रेन के एपिथेलियल सेल्स जो कि सेम वायरल पेप्टाइड एक्सप्रेस कर रहे हैं, उसमें हमें डेथ नहीं मिला. सो दिस शोज कि दूसरी स्ट्रेन है तो MHC मॉलिक्यूल भी दूसरा है. अ वेरी इंटरेस्टिंग फैक्ट इज, MHC को हम हाईली पॉलीमॉर्फिक जीन लोकस कहते हैं, राइट. क्योंकि एमएचसी भी हम डिस्कस करेंगे थोड़ा सा डिटेल में, बट अभी हम पहले एडेप्टिव इम्यून सिस्टम को खत्म करेंगे। फिर हम इनेट में, जहाँ पे एमएचसीज और एंटीजन प्रेजेंटिंग सेल्स, उसका लोकेशन, ऑर्गेनाइजेशन कैसा होता है, वो हम आज से 2-3 क्लास बाद पढ़ेंगे, वहां देखेंगे। पर इतने नंबर ऑफ डिफरेंट जीन्स है जो कि साथ में पॉलीमॉर्फिक, मतलब मल्टीपल जीन लोसाई आर इन्वॉल्व इन एनकोडिंग द सेम प्रोटीन, राइट? तो या फिर जैसे एक के दो एलील्स है या अलग अलग लोकस है, इट्स अ डिफरेंट, लिटिल ट्रिकी टू अंडरस्टैंड कॉन्सेप्ट। अभी आप बस इतना समझो कि इट्स अ हाईली पॉलीमॉर्फिक, मतलब बहुत सारे कॉम्बिनेशंस में एमएचसी बन सकता है। हर इंसान के अंदर एक ही कॉम्बिनेशन का एमएचसी होगा, but the chances of my combination of MHC being the same as the combination of MHC in present in either of you is negligible. So every individual has a signatory MHC complex. ठीक है? तो बेशक H1, H-2K और H-2D is the same species. It's just difference in strain. तो भी टीसीआर recognition of antigen नहीं हो पा रहा है. This fact is known as self MHC restriction.
ओके मैम, एंड मैम अगर यही केस एंटीबॉडी के साथ में होता, सो इन दैट केस एंटीबॉडी उसको रिकॉग्नाइज कर लेता, क्योंकि वहां पर एज सच, क्योंकि सिर्फ एंटीजन नहीं रहता वहां पे,
क्योंकि वहां
एसोसिएटेड
सिर्फ
मॉलिक्यूल्स नहीं है।
एंटीजन है, उसमें एमएचसी का कोई लेना देना नहीं है, राइट?
Okay, ma'am, got that answer. So, ma'am, if that viral is infecting the another cells of our body, so in that case, that particular strain of the CTL will not going to kill that infected cell, right? Because in that case, the MHC molecule will got different, right?
Every cell on the body of the same individual is going to have the same MHC molecule. तो अगर एपिथेलियल सेल की जगह मानो कि हमारा कोई डर्मल सेल है, या फिर कोई भी दूसरा से लिवर सेल इज देयर, या मैक्रोफाज इज देयर, which is presenting viral peptide A to this CTL, there is still going to be a recognition and killing, because every cell of your an individual's body is having the same MHC complex. Got it?
Yes, ma'am. So different individuals में नहीं हो सकता है ये पॉसिबल, बाकी
Yes.
सेम इंडिविजुअल में
Yes.
हो सकता है, बिकॉज उसकी
Yes.
बॉडी में एक ही
Yes.
तरीके का एमएचसी जो है वो सिक्रीट हो रहा है।
Yes.
ओके मैम, गॉट दैट पॉइंट।
Okay. Anything more from anyone? Shall I'll switch the class off and I'll see you all the next.
थैंक यू मैम।